Hello, good morning, everyone.
Good morning.
Good morning.
Good morning.
Yeah,
Morning.
good morning. So you can all see my screen, is that right? Yeah.
Yes.
Yeah, good, good. Fantastic. Okay. Okay, so let's get started. Today we're going back to early vertebrate development, and we're going to talk about. We're just going to compare the mechanisms of development for different vertebrate embryos. Just a reminder that what a lot of development is to model human development, and I just want to show this little movie again because I think it's kind of fun. Just talking about the initial steps in human development, and what we'll try and talk about is how we can use other embryos to somehow understand human development as well. Okay, so I think you get kind of the view. This is a human embryo. We just had a look up to gastrulation stages, and I think that those are the stages I want to talk about today. Let's see. So everyone is comfortable with what's a vertebrate. I think we're all part of the Deuterostome lineage, and we've split off from our tunicate cousins a long time ago with the invention of a notochord, and then from the non-chordate vertebrates, so hagfish, for example, and and lancelets, we've developed a vertebrae. So now we're jawed fish. We have 4 limbs here, tetrapods, and we now have an amnion, a yolk sac, which allows us to lay our young which allows young to be nurtured by yolk. Okay, so this is so we're an offshoot of reptiles, which includes turtles, crocodiles, snakes, and birds. And this is us, mammals. Okay, so the history of vertebrate evolution is kind of all about the innovation of various bits of the vertebrate, things like jaws, things like lungs, things like 4 limbs, tetrapods, and things like the amnion. Okay, and so today what we'll try and compare are the developments of is the formation of looking at development in amphibians, in a fish, in a reptile, and then we'll come back to mammals as well. And so this kind of chart is really nice. It compares some of the major landmarks that happen in early development. And so, like I said, I only want to talk about the earliest developmental stages. So we have, up here we have Xenopus, we've got Drosophila, mouse, cow, human, okay? And you can see here a comparison of timing as well, okay? So Xenopus divides very quickly after fertilization, and within 4 hours you have a very nice blastula. Mouse and human take a little bit longer, and we'll come back to the reasons why this could be. But when we're thinking about these early developmental steps, what we're trying to think about are these things. Okay, so we're trying to remember we're trying to resolve things that are that fertilization has set up. So as I mentioned in one lecture a long time ago, the things that fertilization that happen soon after fertilization is, number one, the completion of meiosis, the fusion of the parental nuclei, the pronuclei, the reprogramming of parental genomes for totipotency, mitosis needs to be reinitiated, and the centrioles come back. And so these are the steps that are fairly common to every embryo. The next thing that should happen is zygotic gene activation, and so the zygotes transcription starts. This is meant to be maternal, not material. Maternal RNA and protein destruction and cleavage as well. And so all of these steps need to happen in when we're thinking about early development. So let's first think about cleavage, okay? And so we talked about cleavage a while ago, and so we have these kind of different types of cleavage. I don't think we need to go into this again, unless anyone wants me to. Does anyone want me to do this? Good. From the silence, I assume you're okay. You all remember very, very well about cleavage. But what we're going to talk about is what dictates cleavage patterns and subsequent development, okay? And so as kind of just so I know that there's someone here and listening to me, can anyone tell me what does dictate cleavage patterns and yeah, what dictates cleavage patterns?
The amount of yolk in the egg
Fantastic.
and how it's positioned.
Yeah, great. Anything else?
The way the centrioles are arranged.
Yeah, fantastic. Yeah, absolutely. Can anyone think of anything else as well that I may not be thinking of? Okay. I can't think of anything else apart from yolk distribution and centriolar distribution as well. The other thing is embryo shape, of course. And so embryos start off, they have kind of very varying shapes, which we'll talk about in a little bit. And of course, yolk distribution. The position of the centriole is sometimes a reflection of the embryo shape as well. And we'll go into this in the next couple of slides. This I'm going to skip through. This is just that amphibian movie that we looked at a few times now, and it's just to talk about cleavage here. And the cleavage of the amphibian embryo is, you can see that it starts off fairly regularly. You have a meridional cleavage, which cleaves the embryo basically in half so that it's along the animal-vegetal axis, and then equatorial, which cleaves then the embryo into animal and vegetal halves, okay? But let's not go into that too much. The other thing we need to do when we're comparing early development is break symmetry as well. And so what is common about early developmental mechanisms are these steps here, what happens after fusion. This step here, the zygotic gene activation, maternal RNA and protein destruction are all common in all embryos. Cleavage, of course, happens in all embryos, but the patterns of cleavage are different. And then finally, symmetry is broken in slightly different ways in different embryos as well. And so we'll talk about this in the next few slides. So this is the amphibian embryo. We've talked about how symmetry is broken here a few times. And so let's just let me see that you're kind of paying attention, not paying attention, that you remember this from when I lectured. Can anyone tell me what's going on here?
The position of sperm entry will determine what is the dorsal side. Yeah,
exactly. Yeah. Thanks, Anandita. Yeah. So you're right. The sperm entry is determining the point opposite the sperm entry point is usually the dorsal side of the amphibian embryo. And the way this happens is that the sperm, when the sperm enters the egg, you have a cortical rotation in the amphibian embryo. And so various components are brought into a new juxtaposition on the dorsal side of the embryo. This is essentially stabilized beta-catenin, and we'll look at this in the next few slides. So what happens is that when the sperm enters, the sperm aster organizes a new set of microtubules, and this kind of tubular skeleton is particularly prominent in the cortex of the embryo here. And what happens then is that the cortex rotates in relation to the core of the embryo. this this rotation brings kind of new components into close juxtaposition. And so what you have normally is this situation in the egg. You have these particular proteins that are forming complexes, and is causing the destruction of free cytoplasmic beta-catenin, essentially. When the cortex rotates, you now have an area where this GSK3 binding protein is able to bind and inhibit GSK3 beta, allowing beta-catenin to become stabilized. Beta-catenin now can travel into the nucleus where it binds to this protein TCF and forms a transcriptional activator to turn on particular genes. Of course, there are no genes to turn on at the moment when this cortex rotates, but once cell division takes place and you start getting nuclei populating this area, then beta-catenin is able to go into those nuclei and set up particular target genes. And so this is quite a simple mechanism for determining dorsal in amphibians. Kind of the thing I find quite unusual is that we generally talk about beta-catenin diffusing into nuclei, but when we're thinking about the steps of amphibian development, it seems like the nuclei are coming to beta-catenin, which is quite funny. The fish develop slightly differently. Sorry, I should ask if you have any questions about amphibian dorsal determination. No? Okay. The fish develops a little bit differently. It has its embryonic part lying on top of the yolk, and division. So cleavage doesn't happen all the way through initially, and cleavage happens just on top of the yolk like this. And so what's essentially going on is this yolk has a hat of embryonic cells, and then the embryonic cells cover then this yolky part here like this. And what we're seeing here is gastrulation taking place. Okay, and slowly you'll start seeing the head of the embryo. The head is here. Somites as the fish starts taking form. Okay, so the mechanisms of symmetry breaking in the fish are kind of similar to the amphibian, and so again you have some symmetry breaking, some cortical rotation that happens in response to sperm entry. This causes enrichment on one side of the embryo of particular dorsal determinants, and even now those dorsal determinants aren't fully understood in fish embryos. It's thought it's probably the same pathway, the beta-catenin pathway, which feeds into, which is downstream of Wnt signaling, and so it's thought that in fish it's a very similar mechanism to amphibians in setting up set in that initial symmetry breaking. It's sperm entry which causes the which causes symmetry breaking and dorsal determination in fish and amphibians. So this is kind of easy. Let's start thinking about slightly other embryos. So these are amniote embryos, and again, those of you who eat eggs know very clearly avian embryology already. You have a very large yolk cell, and every now and then you can see a tiny white patch on top, which is kind of the early vestiges of the embryo. This forms very similar to the fish for some part of it, in that the embryo is on top of the yolk cell. So this is, and we'll go into avian embryos very soon. Marsupials, which are egg-laying mammals, we can quickly go into how marsupials develop as well, and as a comparison to mammals. So mammalian embryos, I think we've all talked about this, form a blastula like this. They have no yolk. Their cleavage is holoblastic. They'll form a blastula like this. And because they have no yolk, there's, of course, there's no topology that we need to think about when we're thinking about yolk. Marsupials also undergo holoblastic cleavage. And so, but what happens here is that the yolk is extruded into the blastocele cavity like this. And the embryo itself then is organized around the yolk, which is kind of extruded from the cells and into the blastocele. And so marsupial embryos at this stage look very similar to eutherians, other mammalian embryos. But let's think about then avian embryogenesis for the next couple of slides. Here. So this is what's going on in reptiles and birds. Okay, the egg is generated, of course, in the ovaries. Fertilization happens, and then this egg passes down this very, very long ovarian tube. In hens, it can be about 80. It's very, very long. And what happens as we go down this tube is that the albumin is added in various parts. The shell membranes are added, and then over here the shell is added to the egg. Okay, and so by the time the egg is laid, already substantial development has happened. But let's talk about the development. Fertilization happens all the way here. Okay, so at quite a distance from the uterus. And so what birds and reptiles have done is set up places that act as sperm storage sites. So there's one here, this primary SSS. There's one here, the secondary SSS. And these will store sperm so that whenever an egg is released, there's sperm there to cause fertilization. One thing that's peculiar about birds, and I think reptile development as well, some reptiles, is that in birds, they seem to use polyspermy to aid fertilization. So fertilization needs multiple sperm to enter the egg. Of course, only one sperm will fertilize with the female. Only one paternal genome will mix with the maternal genome to cause a fertilized embryo. But the other sperm nuclei are kind of retained within the cytoplasm of the oocyte as it divides for some reason, and it's still not clear why this is. During then cleavage and the regular destruction of the maternal components, these nuclei are cleared. And so why this happens is not really understood very well. The cleavage of the embryonic the avian embryo happens as shown down here. Like I said, the one cell is above this huge amount of yolk. It cleaves partially, forms layers, and then organizes into a proper layered embryo like this. We'll see this in the next slide, I think, here. And so this is development of the avian embryo. You have multiple cleavages that happen in waves that go from this single cleavage plane. Another cleavage plane happens here, and so on until we have many cells forming many layers. I'm going to use the avian embryo to talk a little bit about zygotic gene activation as well, which has been worked on in all species, but I'll talk about it here. Okay, so zygotic gene activation is, of course, when when transcription in the zygote starts. There are two ZGAs, zygotic genome activation. There's a minor and a major ZGA, and so all animals have this. A minor ZGA is some of the zygotic genes are activated, not all of them, but just a very small percentage, and this percentage can range from about 5 to 20% of genes are activated, and these are activated very early. The major ZGA activates almost all the genes, so all genes are activated at major ZGA. All zygotic genes that need to be activated are activated then, and at the same time, the maternal RNA and protein components are destroyed as well. So these the timings of these kind of vary a lot, and you can see here in the mouse, for example, minor and major ZGAs essentially complete by the second division. In frogs, the fourth division is when you get the minor ZGA, and by the 14th division, the major ZGA occurs. And so this disparity, I'm sure, reflects the ecology of the embryo and how where it's laid, what events it needs to, what events have provided some evolutionary pressure to the embryo, and so the timings of, the difference in timings of major and minor ZGA between different species is still unclear. The mechanisms are known about on what triggers ZGA, but what the evolutionary pressures are that change the timings in different species is not so clear. Okay.
Raj, what is EGK?
EGK, thank you. Sorry. Thanks, Shubha. This is so what you're looking at here is the avian embryo. avian embryos, like I said, avian embryos develop outside, have a substantial amount of development going on inside the egg, inside the head. And those stages of development are shown by this stage series, the EGK series, which is Galadi stage series. And so this is the EGK stage 3, means this, it's about 10 or 12 divisions. EGK 5 is this many divisions, for example. And so that's all it is. It's a stage series. Okay. Galadi. Is that clear enough?
So it's the stages of development that are taking place inside the parental body.
Yeah, inside the head. Yeah, exactly. Yeah. I'm sorry, I'm a little bit. So I'm lecturing from London at the moment. It's 4 a.m. here, and the coffee is just now kicking in. Okay, so I'll make sure I am awake much earlier than the lecture next time. Okay, so going back to the development of birds, and reptiles, in fact, is the symmetry breaking events that happen in birds. And here, once we get to amniote animals, so reptiles and mammals, essentially, the symmetry breaking events are not so clear, and it seems like it's almost random which side gets called anterior, which side gets called posterior, and minor fluctuations in conditions seem to influence the symmetry breaking events in these animals. And so, for example, in the avian embryo, as the egg passes through the hen, it rotates, and minor tilts in the egg cause different juxtapositions of interembryonic components. Here, this tilt, for example, of this egg at this stage causes then the yolk density axis to shift in comparison to the blastodome yolk axis, which then sets up an asymmetry very similar to cortical rotation in the amphibian. This then is reflected by some idea of lineage segregation. And so what Guozhen Sheng suggests here is that there are already cells that are specified as anterior and posterior mixed within the embryo. And these kind of asymmetries here just help these cells to sort out along the axis. And so essentially what symmetry breaking is doing is allowing sorting to happen within the avian embryo. This is for me, it's a slightly conceptually difficult thing to think about, that anterior and posterior cells are existing in kind of a random salt and pepper method, and then they sort out according to perhaps some gradient that's set up by the yolk density axis. And so, but because these early embryos, these early avian embryos are very difficult to get at because they're developing within the hen, it's kind of still very difficult to understand what's going on. You do have many asymmetries happening within the avian embryo. Like I said, so these borders here where the embryo touches the yolk form syncytia, So these cells are contiguous with the yolk cells, and so you can imagine that if there is kind of this slight enrichment of yolk on one side than the other, we'll start getting asymmetries, maybe even metabolic asymmetries happening across the embryo, which may feed into patterning the embryo. It seems like a sloppy mechanism, but maybe it's okay to be sloppy as long as afterwards the embryo regulates and knows to at least form an embryo that has a front and a back. Okay. There are similarities between the avian and mammalian embryo as well. And these are not immediately apparent because the avian embryo does develop to some extent within the hen. And so this is actually one of my papers, where what we looked at was two different bird embryos, the finch embryo and the chick embryo, and we found that oviposition of finches, so oviposition meaning when the egg is laid, the finch embryo is much earlier than the chick would be at oviposition as well. And this this gave us an insight into what's happening in avian embryos at this kind of stage versus this stage. And we were able to, for example, derive stem cells from the finch embryo, ES cells from the finch embryo, but not from the chick embryo at these stages, just because the chick has developed more in the hen than the finch has. Okay, so that's avian. We'll finish up with mammals. So here we're looking at mouse embryos. You're seeing division occurring kind of in different planes. And let's look at this cleavage in more detail. So as someone mentioned, as Grish mentioned, the centriole position does play a role in determining cleavage plane. What centriole position essentially does is also set up asymmetries within the embryo, and these asymmetries consist of these particular proteins here, the PAR complex, Rho proteins, and so on. The name of the genes aren't so important, I think. And these asymmetries feed into the cleavage plane, the patterns of cleavage for these various embryos. Let's go to mouse. The mouse embryo, so the mouse oocyte, when it's laid, has a asymmetry. As you know, the nucleus is on one side and not the other. So sorry, the meiotic nucleus is on one side, not the other. This is the side that's closest to the pronucleus is called the animal side, and the other is the vegetal side. And this side is characterized by the expression of these particular proteins, the PAR3, PAR6 proteins. This seems, and so soon after fertilization, this kind of asymmetry disappears, and so you no longer see this animal-vegetal distinction once fertilization is going on. But it is important, and I'll show this in the next couple of slides. Soon, so division happens for about 2 or 3 cleavages, and after that you start seeing the appearance of particular epithelial polarity genes again. So PAR3, PAR6 will start being expressed in the outermost cells. The innermost cells seem to have a basolateral character. And this resolves as development goes on. And we'll talk about this in the next slide. So, as I mentioned, mouse and mammals in general have a kind of two different modes of cleavage, and so cleavage can be meridional. So if you can anyone remind me what meridional means? Oops. Okay. So meridional cleavage the cleavage plane is here, okay? This way. Can you see my arrow?
Yes.
Yes.
Yeah, good. Thank you. Yeah, so the meridional cleavage is like this, okay? Equatorial cleavage is like this, okay? And so when the mouse cleaves meridionally, this animal and vegetal are equally distributed to both cells. When the embryo cleaves equatorially, one cell will inherit the animal and the other cell inherit the vegetal poles. Now remember, after fertilization, there's no real overt difference between animal and vegetal. So these PAR3, PAR6 proteins are no longer asymmetrically expressed after fertilization. Yet there seems to be some, when looking at kind of the progeny of these kind of cleavages, there seems to be some difference. And we'll go into this in the next slide. So it seems that in most cases, you have the first cleavage is meridional, and then the next cleavage is equatorial. And this gives rise to this kind of embryo here, where much of the inner cell mass, so the embryo proper, is derived from these meridional cells, and the equatorial cells then give rise predominantly to the trophoblast. Even when the first cleavage is equatorial and then the second one is meridional, we get the same kind of distribution. If we have two meridional cleavages, then the cells are kind of are distributed throughout the lineage. So what these cells will give rise to is distributed throughout the embryo. But when there's two equatorial cleavages, then viability of the resulting embryo is much reduced. And so again, what these cytoplasmic determinants are within the mouse and presumably the human as well, that are being inherited by these cleavages is unclear. There's no overt polarity to the embryo at this point, but it does seem that the pattern of cleavage for the mouse makes not a huge, well, a fairly significant difference to the viability of the resulting embryos. So do we have are there any questions to this point, up until this point? Okay, cool. So the final slide, I think this is the final slide, is well, penultimate slide, is comparing then models of mouse development. And like I said, what we're trying to do what one aim is to understand human development. Mice actually look different from most mammals. And so while the embryo starts off looking kind of the same, so here, this is kind of early implantation embryos here. You're seeing the blastocyst forming, which looks very similar to the human. As it implants, you're seeing this kind of what's called the epiblast rosette forming here. At implantation, the mouse embryo looks very different. It forms a cylinder, and so the embryo here in yellow is organized as a cylinder, as a cup, and development happens within the cup. Okay, all other mammals essentially look like a disc, so they look like a chicken essentially. The embryo is here, and you're looking onto the dorsal surface of the embryo when you're looking up here. There's the streak is forming down here, and you're forming mesoderm over here. And so, in some respect, the mouse is not such a good model for human development. It looks different when it develops. Things like the primitive germ cells are in different places in the mouse than they are in the human, and we have kind of this trophoblast which covers the human embryo. Is not so distinct within the mouse. This huge covering that nourishes the human embryo and other embryos as well has been converted to this simple ectoplacental cone in the mouse. Some people say that, so there's some theories why this kind of conversion could happen. Rodents are, of course, one of the most successful mammals, and they're successful because they do have many offspring, and are able to, the females are able to carry huge litters and multiple litters during their adult life. And it could be that this is an adaptation to pack more embryos into the uterus of the rodent. So this is one major difference, and so much so that probably the best mammalian model for human development is probably the rabbit. And so here, what we're looking at is the development of the rabbit embryo. And you can again see cleavage happening, the enlarging blastocyst happening at E4, and then by implantation, gastrula stage, you can see very clearly now this is the embryonic disc, and you can see the streak starting to form here as well. And so this is very much similar to what's going on in the human. Okay. So that's really all I have today. If there's any questions, I'm happy to take them now. Any questions? Cool. So we will come back on Wednesday and talk about kind of comparing gastrulation and thinking about gastrulation in all of these vertebrate embryos. And then on Friday, we'll talk about mesoderm development as well. I kind of want to start talking about the evaluations for this course. And so, so before I move on to that, if there's any questions about this particular lecture, let me know. So you can tell me now, or if you think of anything, send me a message through Moodle or through my email address. The evaluation, so I'll talk about the evaluation then. So the course will be, the evaluation for the course, the end of course test is essentially an essay. Yeah, all I'll ask you to do is to write one of, is an essay on some aspect of development that you find interesting. So, and this needs to be kind of a well-researched, well-referenced essay. There's a long timeline for when it should be done by. So I'm thinking sometime in the beginning of February, we'd want the essay, and that will be the majority of your mark for this course, okay? So this is kind of a starting pistol. So you've heard me lecture, you've heard Tina lecture. From next week, you'll have Sonia and Bhavna lecture you. And so within those particular topics, or any other topic that you find exciting and interesting, that has some aspect of development. And so remember, my definition of development is quite broad. So if any of those, if any part of that excites you, start researching it and start laying down some ideas for your end of term paper, end of course paper, okay? Great. So
how long should the essay be? Like how many words or anything like that?
Yeah, it doesn't need to be too long. Shouldn't be too short here. Sorry, so generally I would say about 1,000, 2,000 words. I'm happy to accept up to 4,000 words if you feel that you have much more to say. But again, it needs to be a complete review or of an idea. It needs to present some synthesis of existing data, present some new idea that you may want to present. And so the word limit is not so tight, I think, okay? As long as you can put something something good down, okay? But I'll talk to Tina and set this up in a much more formal way, okay? Cool. Okay. Yeah, Grish.
Could we also write it on plants?
Yeah, sure.
Okay.
Yeah. So if you do write it on plants, I'll get Shiva to look at it. Okay. And so what I will do is whatever you want to write it on. So if someone wants to write on bacteria and single cell development, I'll ask someone who's an expert on this to take a look at it. If you want to do hematopoietic development and things like that, we'll try and give it to various faculty who have more expertise in this than I do. Okay.
Yeah, thank you.
Cool. Great. Okay. So final thing is, sorry, was someone about to say something?
I had a suggestion. I mean, I was wondering if we could all decide and meet at a later time if that's more convenient for you. At 4 a.m. We don't want our lectures going crazy.
So 4 a.m. sorry, it's okay. For me, it's okay. I think I'm this is the first time lecturing after a slightly long absence. Yeah, it's okay.
Okay.
This is fine. Yeah, it's all right. I'll go back to sleep. Okay, but thank you very much. Okay, so that's all. If I think some students wanted to talk to me about other things. I can if it's okay, I can do that now. And so anyone who wants to talk about physical joining or any other concerns that they might have, we can talk briefly about that now, if that's okay. Or if you want to set it up for another day, we can do that as well. Any? Akash.
Hey Raj. I was asking that you told Shubha that we have to join campus by 5th January, right?
Yeah.
So would we be getting some formal mail regarding that, or it's just this way?
No, you should have got the formal mail a couple of weeks ago, and so I need to follow up with the academic office about this. Okay.
Okay, because I don't think we got any mail regarding that.
Okay. So I'll follow that up. Okay. So the plan for you, for all of you, is to so Shubha, you sent. Did you talked about this to the rest of the batch? Did you? Okay.
She did tell us.
I just told them just as what you replied in the mail.
Okay.
I just told them that by 4th the current plan is to come on 4th.
Yeah. And did you? I did send the student handbook afterwards. Did you all receive that as well?
Yeah.
Yeah.
Yes.
So some of you who aren't in the, so Ayanthi, did you receive a student handbook as well?
No.
Okay. So maybe the email address doesn't capture everyone.
Yes.
Yeah, I can send that. Okay, so the idea is that you're here by the 4th of January. I will ask Vishalakshi and Anusha in the academic office to make sure that they send that message to you. I also had the student handbook that they should have sent out to you as well. And so which I've sent now. And so that will get done. The idea at the moment is to, once you're here, you'll need to go into quarantine, I think, for 5 days. You will get tested after 5 days. And when you're with your negative test, you'll be moved into the new hostel. Okay, so you guys are going to be the guinea pigs for the new hostel. Is that okay? Yes, Garish.
Are we going to stay there for 1 year or the rest of
the rest of your tenure?
Okay.
So I don't think you'll move around. Okay.
Okay. Yeah.
Okay.
So those coming in from Bangalore, so some of us are in Bangalore. Should we also quarantine when we come to campus?
No. You don't have to quarantine. Yeah.
Okay.
So Shubha and Sahana. Yeah.
Yeah.
Yeah. So yeah, you don't have to quarantine. I mean, you guys just live down the road, yeah. So you can communicate with your batchmates outside their hostel. There were some other questions that you had. And I can't remember what they were. Are there any other questions that anyone has for me?
I had a question. What would be the food and the other essentials during the quarantine and all? Like, would we be provided with it, or we have to take care of it ourselves?
So food you'll be provided with, okay? So you'll be given 3 meals a day for the 5 days that you're going to be in quarantine, and the other essentials. So for the hostel room, and so this is going to be a little bit. I don't know how we can work this out. The idea for the hostel room is that you provide your own bedding, okay? So you provide your own mattress and other bedding, okay? And so I think you'll need to bring that. Yeah, either you bring it or you buy it once you're here. And so that's kind of one of the main things that I think you'll need to do. I'm still trying to work out whether we can deliver your, the majority of your luggage and bedding and things to your hostel room. And then in your quarantine room, because they're 2 separate places, you can have whatever you need for the 5 or 6 days. Yeah. So we'll work that out. Okay. Okay.
All right.
Yeah.
Thank you.
Yeah. But I should say that whenever you want to get in touch with me and chat to me, yeah, feel free. Yeah, I'm not doing much. Yeah, I'm just waiting to look after you all. Okay, so feel free to get in touch. The other point that Shubha raised was about rotations and PIs. And do you want to discuss that now, or does anyone have any questions about that?
So what we thought was the labs that are being presented at Open Labs, we thought that they were the ones which are going to accept students. But then on Thursday, we had an open lab, I think by Dimple, and then she said she's not going to accept. She doesn't, because she wants certain students in her lab to finish their thesis, and only then she'll take in new students.
Yeah. Yeah. So again, and so then Shubha, I did send you. I think I sent everyone the faculty list. Is that right?
Yes, we got that.
Yeah. Yeah. And so that again should have been sent to you, sent to all of you a long time ago. I suspect that. So there was a problem with the batch email initially, which I think, which meant that things weren't being sent, and maybe this got lost in there. But the Open Labs, yeah. So some of the Open Lab presenters are not taking students, but the idea was also then to see what everyone is doing, and whether if they're not going to take a student, whether at least they can possibly be a TCM member, or whether you can use their expertise for something that you want to do. Okay.
Are they taking rotation students or not at all?
They may take rotation students. It depends on the PI. But also, I probably would recommend against rotating in a lab that is not taking a student proper.
Okay.
You don't really want to waste your rotations. Okay. Yeah.
Thank you.
Yeah.
Raj, from which month will the rotation start?
They'll start from they'll start towards the end of January. Okay, so the idea at the moment is that you will be in your hostel by about the 12th of January. We'll have a kind of a follow-up of, we'll have a an in-person orientation at that point. And then give you a chance to kind of maybe a week or a week to talk to the various PIs one to one, and then we'll have this kind of session, kind of rotation counseling session after that. And then my idea is that by the last week of January, you should be rotating, okay? So the rotations are 6 weeks long.
Yeah, okay.
Okay. Hi, Girish.
Raj, when are the annual talks starting?
The annual talks will start on the 7th of January.
And we'll be attending that, and we won't have courses during that time.
You won't have courses during that time. You will be attending it, but I think all of the annual talks are essentially online, okay, this year. So I think you'll probably be in quarantine for 2 days of the annual talks. And so you can attend them online, and then when you're out, I think the last day you can kind of go in person and see the posters. Okay.
Yeah. And
Okay.
when will the next course semester start?
The next course semester should start in the week after the annual talks. I just need to push and prod the faculty to give me their course details. Okay.
Then when we are submitting at least the end term paper for this course, we'll be also attending other courses.
Yeah. So it's good to just get this one polished off while you can, while there's not much going on. Okay.
Okay. Yeah.
Also, Raj, we were receiving some mails about maybe a seminar or a workshop on lab ethics and safety rules. Yeah, could you maybe tell us more about that? Yeah, I guess that's compulsory for us.
Yeah. So once you're here, the lab support run a mandatory safety course. Without that, your ID cards are not active. And at the same time, I'll do a session on kind of in research integrity as well. There's not much to tell. I mean, it just tells you which fire extinguishers to use if you cause the fire, what you should do in various contingencies if something goes wrong. And my part is to talk about what is academic and research integrity. Okay.
So we'll be attending those after we reach campus.
Yes. Yeah. Although everything is online at the moment, but I think it's what I don't think you need to worry about it at the moment, because I mean, yeah, it's going to be a prelude to doing your rotations. Okay, so during the in-person orientation, we'll try and do it then. Okay.
Okay, thanks.
Yeah. Okay. Cool. So any other questions? So so I'm happily lecturing you for the next two developmental biology lectures. And so if you think of any questions, not just about developmental biology, but about being a student on campus, then bring it up after the lecture, okay? Yeah?
Yeah.
Okay. Super. Okay,
Thank
everyone.
you, Raj.
Thanks. Take care, okay? Don't do connect with me if you have any concerns or doubts, okay? I'm fine to talk to you and listen to them, okay? Yeah.
Thank you, Raj.
Thank you.
Okay.
Thank you, Raj.
Thank you.
Thank
Take
you.
care,
Thank you.
everyone.
Thank you.
Okay.
Thank you.
Take care.
Bye.
Bye.